back. It is my enormous pleasure to introduce today Craig Venter, who is someone I feel almost needs no introduction, but I will attempt one anyway. So Craig is a true pioneer scientist and entrepreneur. He is a giant in genomics. He was one of the first people to sequence the human genome. He has explored the metagenomes of our ocean microbial communities. He is also a pioneer in synthetic biology, having synthesized the first synthetic biological organism. And his latest venture is Human Longevity, Inc., a company focused on extending healthy, high-performance human lifespan. So just to get things started, how many of you have had your genome sequenced? Okay. How many of you are interested in getting your genome sequenced, but you're not sure what you're going to learn from it? Okay, your work is cut out for you.
It's a good business opportunity.
Yeah. So tell us a little bit about Human Longevity, Inc. What are you doing, and what do you hope to do?
So human longevity is a basis of what we started a long time ago. So June was the 20th anniversary of us sequencing the first genome in history, a small bacteria, developing new algorithms to make it all go together. 15 years ago, in fact, I was with President Clinton yesterday at the Clinton Global Initiative, celebrating on CNN our announcement in the White House 15 years ago of the first human genome sequence. But that genome cost $100 million, took 9 months to do, and wasn't going to be highly replicable for a while. So basically, since then, I've been waiting for the technology to change, the computational tools to change, so we could start the real experiment and the real analysis where we need hundreds of thousands to millions of genomes, all with phenotype information together. So with the advent of the Illumina X10 sequencer, sequencing just crossed the threshold that we were looking for. So we sequenced genomes for about $1,000 each. Computing was the biggest issue. We built the third largest computer in the world 15 years ago. It cost $50 million and was only 1.5 teraflops. So you can buy a card for your PC for about $100 for that now. But with cloud computing and distributed computing, there's the potential for computing to stay ahead of the needs of this space. But we're already busting through the ceiling of Amazon cloud computing. We needed to do a recompute on just 10,000 genomes, and we needed 4,000 or 5,000 extra processors, and they couldn't instantly provide them. So people think of this as not really a big data problem. HLI is in the top 1% of Amazon users, but the biggest users are really large because they're mostly streaming videos. But it's not complicated calculations. You don't need lots of processors to stream videos. So that's what much of the cloud is used for. When you're trying to do huge calculations with distributed computing, you need lots of processors. So it's on the verge of being there, but if we're having trouble with tens of thousands of genomes, millions of genomes, we need the compute world to accelerate a little bit more. And the third component is machine learning. So I was a real fan of what Franz Ock did at Google, creating Google Translate. And I thought it was a good paradigm that would be useful for understanding the genome. So with some difficulty, I recruited him out of Google, and he heads up our machine learning group in Mountain View. So we're putting machine learning together, really advanced distributed computing, and very high throughput genomics. We've already done about 20,000 human genomes, all with phenotype and clinical information across lots of disease areas. And we're scaling up very rapidly. The goal is to create a database for understanding this information, not assaying it just as a diagnostic. And the machine learning is already giving us some exciting data, even with 20,000 genomes. But we're excited about getting to the next stages.
So what kinds of things can we learn now, with the state-of-the-art genomics with respect to human health? And what are some of the things that you hope to achieve, with your database?
Well, just changes in what was possible with the old school tools in genetics that pretty much peaked 20 years ago, where you needed large pedigrees, lots of time to try and get some kind of linkage to link things to some part of the genome. Now, from a single genome, we can solve genetic disorders. We brought a young child from Children's Hospital with the diagnosis of Delleman syndrome. There's only 30 cases in the world. And it's unusual with lots of tumors covering the body, but a very misdeveloped brain, basically scrambling of much of the brain. And nobody had any understanding of the genetics with this. With this, we can simply sequence the whole genome of that individual. We did a trio. We also did his parents. And it became immediately visible what some of the problems were. It was obvious from their genomes that the parents were very closely related. It turns out they were second cousins, but from a small village in Mexico with lots of inbreeding. So they had some whole chromosome arms that were virtually identical. So that's certainly a way to have penetrance of traits and changes that you wouldn't normally have. And we found 5 genes associated with neuronal development that were badly mutated. And then we found a mutation in the neurofibromatosis 1 gene, but it was interesting because neither parent had that mutation. So it was clearly a spontaneous mutation. So you can see the rareness of it. It's a multigenic disease, 5 genes associated with neurodevelopment plus neurofibromatosis that explain the different sets of symptoms, all with one individual plus his parents. And so it's the power of this, but our collaborators at UCSD said even with 2 years with using the UCSD supercomputer, they couldn't have gotten to the same answer that you can get when you have a 20,000 genome database to understand allele frequencies, what's meaningful and what's not. And this is the earliest stages. As we get 1 million genomes, the power of all this will change dramatically. But the multigenic part of it is, I think, the main thing we hope to come out of this type of study. And with machine learning, we look across the genome. There's no hypothesis that goes in that it might be this gene or that gene. And we've certainly had to train the entire team to not think in terms of single SNPs, single gene alleles, because that's a limitation that comes out of what the genetics community has been doing. And machine learning just looks across the genome and finds all kinds of associations. So the task we gave to the machine learning group initially was to see if we could predict your photograph and your voice from the genome and finding correlations between them. And it's at the stage now, if we had 100 genomes and 100 photos, we could easily tell you who was who. We can predict eye color better than you can predict your own eye color. The measurements are getting extremely interesting. I was more surprised by voice and just what you can predict from voice alone. So from a voice recording, we can accurately predict your sex, your age, and your height. And some of these correlate with face shape and alleles associated with that. So we look at humans as an integrated unit, not just as a heart or a tumor or some other aspect. And all these things are interrelated. I never would have looked for the shape of the face as being part of the voice, but when you learn that it is, it's not illogical that it is. So those are the kinds of associations we're making at the moment.
So how much harder is the problem of solving something such as healthy aging than predicting your face shape?
Yeah. I think healthy aging's going to be a lot simpler, probably, than those kind of correlations. So maybe this is not the right group. There's a few slightly older people here, but if you're between 50 and 75 and you're a male, you have a 30% chance of dying during that time period in the US. If you're a female, you only have a 20% chance of dying during that time period. The numbers are a little bit different from males and females. Females have a higher risk of dying from cancer than from heart disease. Males, it's roughly equal cancer and heart disease. So you can affect healthy longevity quite dramatically if you can just predict risk for cancer and heart disease that can become actionable. So we're certainly focused on that as a major focus initially, but as I said, we're focused across the entire genome. I think Larry Page told me that if you eliminated all cancer, you only change the average lifespan by 3 or 4 years. But that's the fallacy of averages. That doesn't mean anything at all. If you're a 6-month-old kid and you die from neuroblastoma versus Jimmy Carter getting melanoma in his 90s, those averages are very different in terms of the real outcomes. And so getting real numbers, real data about each of us is what the goal is.
And so how are you going about acquiring this data, and ensuring that it's of the quality that is going to be useful to you?
So that's one of the biggest efforts we have going on right now. We have teams that are collecting great clinical cohorts from around the world. Some examples that might be familiar to some of you, we have, there's a 20-year identical twin study out of the UK that people have been measuring things on these twins extensively for the past 20 years. And so simply in exchange for sequencing their genomes, we get all 20 years of phenotype data to try and make correlations with. And the fact that they're identical twins is great. But phenotype data is very variable with every group and study. We had to send a scientist over to the UK for 3 months to help sort out and deconvolute some of the data. But we get our best data from clinical trials. We have a great collaboration with Genentech and Roche. They were the first ones to sign up to work with HLI, and we're doing about 10,000 genomes a year from their clinical trials. And clinical trials, the data has to be very standardized in what you measure, so we get very clean phenotype data that's easy to deal with from Genentech. In exchange, we give them very clean, very accurate DNA sequence. And the trick is trying to put this together. But that's what we do. We won't do any sequencing. We don't do contract sequencing. We'll only exchange genomes, sometimes for a fee, sometimes not, for the good phenotype and clinical data. So we have literally hundreds and hundreds of collaborations around the world. We hunt around for the best phenotype characterized populations. In exchange for getting the genome, we get that data. And that's what's really enriching the database quite tremendously. And we're negotiating right now with 8 big pharma companies that all want to add tremendously to this. And last week, we just announced our first major insurance deal with Discovery Health in South Africa and in England. They have 4.4 million clients that they're going to add genomes to. And we announced a unique product of the lowest price exome. So we do the entire exome for $250 for these clients. In exchange, we get all their phenotype data and their lifelong outcome data out of the database. So genomics is starting to penetrate deeply into changing how clinical trials are done. And I think it's going to totally transform the insurance industry very rapidly. But we're trying to have the richest source of all diseases as we can. So we're not focused on any one area. The database has to be across traits and across diseases. We have a special project collecting extreme phenotypes. So if any of you have a really extreme phenotype, let us know. We'd like to characterize that phenotype and do your genome. For example,
maybe for the people who aren't totally familiar with the science, what is an extreme phenotype? Give us an example.
Well, one example is we have a large population out of the highest altitudes in Peru. You'd think everybody that lives in these high altitudes are totally accustomed to high altitudes. But a certain percentage of these populations get altitude sickness and simply cannot survive and thrive there. So the extreme phenotype, we're looking at the genomes of the ones that thrive at high altitude and compare them to the ones that don't, so we can predict readily just from the genome whether you could survive and thrive at very high altitudes. And maybe that'll make you a better marathon runner in the future.
So the goal of this database really is to collect, we talked about this earlier, 1 million genomes with very, very rich phenotypic data about anything that you can possibly measure or characterize about people. And then you're going to use machine learning to draw relationships between all these various axes of your data. Is that right?
So the goal is to interpret everything that's interpretable, predict everything that's predictable from the genetic code. So if we can accurately measure a phenotype, we can find out if there's a genotype that correlates with that. For example, on the 13th of October, we open officially our health nucleus, which is where we do the most comprehensive phenotyping on each individual. We start with sequencing the entire genome. We sequence your microbiome. We measure 2,400 chemicals in the bloodstream with mass spec, and that's before you get there. When you arrive, we do quantitative MRI imaging on brain scans and then quantitative whole body MRI imaging. We do DEXA scan for bone density, peripheral and visceral fat. We have the latest 4D color echocardiogram, so we can actually look at your heart valves and actually make a colored movie of them. We measure everything we can measure non-invasively in an 8-hour period and link that back to the genetic code. So if you want to learn more about yourself than you can learn in any other one place, you should sign up for the Health Nucleus. It costs $25,000, so information is valuable. But we're using this to build the genotype-phenotype correlations in the database. But we have the latest GE3T MRI system that only our system and ones in GE Research have these protocols for measuring unique phenotypes. So anything we can measure precisely in body structures from these images, we can then take into machine learning and find the correlations.
And so who is going to be the first beneficiary of this information about these correlations? So who's going to find this the most useful in the shortest term?
Well, I think the people that are going through it are going to find it extremely useful. Some of you have had some kind of genome analysis, but genome data without the phenotype data and making these correlations is very difficult. We had a number of PharmD students working as interns over the summer trying to do further annotation for pharmacogenomics. And at the end of the summer, we asked them as a group, what's the number one thing they learned? They said they learned the scientific literature is full of crap and is completely unreliable. And that's a very important message because that's in fact the problem with genetic association data. There's so much useless or wrong information in the genome literature that you can't rely on it, and that's why there's such a problem with the FDA. You can't just say, well, this was in the literature, therefore it's a valid correlation. We're trying to sort out what's valid and what's not, and we constantly measure the frequencies in our population now of 20,000, but once we have millions, that data will keep getting more and more accurate. Predicting whether you're increased risk for cancer is very important. Tom Caskey at Baylor did a study on a number of YPO members just doing whole exome sequencing and discovered about a third of them had significant risk for different diseases that most of them were completely unaware of. So don't be disappointed if your genome says you're not at high risk, but at least a third of you will be for something. My genome was the first one that was sequenced. And from all the analysis being done, I learned that I was at high risk for melanoma. I'm in fact in the 93rd percentile for that risk. That doesn't include behavior. You mentioned I sailed around the world. I spent a lot of time, when possible, in the ocean or in the sun. So those must be genetic alleles for risk-taking or liking the sun, which probably puts me in the 300 or 400 percentile for melanoma risk, not just measured by these other factors. And so I learned, because of that risk, to identify melanomas relatively early. 10 years ago, I found one, and it was simply a matter of losing a pound of flesh to get rid of it, because I recognized it early, being aware of it and of that risk. Had I let it go 6 months longer and it penetrated deeper into the dermis, it would have been a totally different situation. The insurance cost would have been extremely high, and if I tried to pretend it wasn't there and let it go longer, we might not be having this conversation. So individual knowledge is power for individuals and makes you independent of the medical system for your own health and survival.
So the questions are coming in. So first one is from Rusha Modi, who asks, when will this technology achieve the scale to be widely available at lower cost?
So how much lower cost do you want than $250? How widely available does it need to be? I think what's going to happen over this next decade is we'll get better and better and better at interpreting the information and getting orders of magnitude more valuable information back to individuals from these large data sets and interpreting it. So right now, the $250 exome is available in England and in South Africa through Discovery Health, but we're in discussion with other insurance carriers. And in the US, most corporations self-insure. So we're in discussions with several corporations where they're considering having the genomes, or at least the exomes, done of every single employee, trying to understand the health risk to do what Discovery Health does, a preventative health, preventative medicine paradigm. So Discovery Health lowers the premium for people that follow preventative medicine paradigms. At age 65, the ones that follow their advice have 8 years average longer longevity than the ones that pay the highest premiums because they don't follow the advice. So it's a new model.
Is behavior part of the phenotyping that you're, is this behavior, these aspects of behavior part of the phenotypes you're collecting?
It could be behavior as well. Obviously, smoking is a deleterious behavior, but what Discovery Health does, they give you points if you go to a gym, they give you points if you buy the right food, get different types of checkups. And so it's standard checking information, but we're adding to it predictions for cancer risk and cardiovascular risk, for example, that they think will even lower those premiums more, but at the same time saving themselves lots of money. So we're trying to bring the paradigm to the US in a form that's compatible with I think the good kind of thinking the FDA has, and wanting to have this done and getting solid information, meaningful information to people, not data with lots of false positives and false negatives.
So we have a question about data access from Narinder Singh. Do you see sharing the data more directly in the future to serve as a platform for innovation, or do you see yourself as being more focused on being a provider of the insight about the data?
So it's actually going to be both. We're trying to build a database that everybody will be able to use as a reference to make interpretations. So pediatricians, if they get a sequence and they're worried about Delman syndrome, will simply log onto the database to compare paradigms. So we're setting it up so pharmaceutical companies like Genentech will be able to just have an annual subscription to it. Same with insurance companies, but physicians and scientists will just log on to ask a single question or more complex queries. So it's only useful if the data is broadly accessible. So the HIPAA regulations right now in the US say you can de-identify a human genome if there's no personal information, name, address, social security number, or a photograph associated with it. To me, it's always seemed a silly notion to claim you can de-identify the information that gives you your identity. And in fact, if we can predict your face and your voice and lots of other of your characteristics, there's no way to de-identify it. And so the data can't just freely float around on the internet. It has to be in highly secure databases. And if we have your genome, we have your phenotype, your clinical records, we're setting it up so nobody can search and try and find you in the database to find out things about you. They can look across the genomes and across the phenotypes to find things in common. So data security is going to become very, very important with this. And if I was giving a younger Craig Venter advice, I'd say don't put your genome on the internet. But I also published an autobiography, and that probably revealed even more than my genome did. So I'd say don't do autobiographies either.
So on the topic of advice, I have a question, a more general question, which is, what has been the hardest part of your career? How have you dealt with naysayers, and what can other innovators learn from your experience?
No, it is an important question. It's you have to have courage of your own convictions. You have to believe what you're doing and what your teams are doing can really work. Most major advances in science and in life come from people bucking the norm of the system. It doesn't always work, but had our techniques not worked, it would have been one of the biggest public blowouts in history. With the Drosophila genome, we finished the calculation a few hours before going to the meeting to present it. Had it failed, it would have been very clear cut. The same with the human genomes. Our data's stood up to the toughest scrutiny. If our data was not perfect, we would not be going forward now. So I like winning arguments with data. So you can buck the system, but you can't do it in a sloppy way. You have to have rigor behind it. You have to have discipline, and you have to have damn good data. And but with that, you can win lots of arguments. So
where do you see us being in terms of, what is the word exactly again? Healthy, high-functioning lifespan in 10 years' time?
Well, if we switch to preventative medicine, as I was able to do in my own case, and I also started taking statins based on seeing my genome 15 years ago, we can change the period in our lives where we have good health and can have active, productive lives. Our goal is not just to have people live longer. It's to, even if the number doesn't change, if you can get up to that end period and be mentally functional, physically functional and active, that's a pretty good deal, and that's a pretty good outcome. There's actually social issues with extending human life when we're depleting the Earth's resources at the way we're going. So I think it would be socially irresponsible to try and help people live to 200 right now, unless we castrated all of them, or just a few of them.
All right, let's thank Craig Venter for a very, on that note.